So you're trying to be a lean, mean workout machine, and you be like eating lots of chicken, steaks, eggs, cheese, milk, and you're trying to be like, yeah, I'm gonna be really strong and lean, and I'm gonna be able to rock it like Rocky Balboa. Well, you may not know who Rocky Balboa is, but anyways, did you also know that vegetarians and vegans can also be like lean, mean workout machines? You'd be like, what? Are you serious? That can't be true because I'll be eating all that real proteins while you be eating like cellulose. Yeah? No, wait. It turns out plants also have proteins. And so it doesn't really matter what kind of stuff you put into your body because, well, it kind of matters, but protein-wise, it doesn't really matter because when you take in all those proteins, your body is going to break it down to its essential amino acids for nutrient up intake into your body. So it doesn't really matter how you get your protein source, as long as you get all the essential 20 amino acids in your body, you are fine. Now, our body also will make some amino acids, but there will be some amino acids where we have to take in. Anyway, there is a whole different topic on nutrition and food intake that I'm not going to deal with because I'm not an expert in those things. So, proteins. This is the basic structure of proteins. You will have a central carbon atom with an H always bonded to that central carbon atom. And depending on which literature you read or which websites you go to, you will see that an amino group is attached to that central carbon atom, and a carboxylic acid group is also attached to that central carbon atom. And the fourth bond that is attached to that central carbon atom is what we call the R group. Now, this R group can be anything. If it's a simple H, then this amino acid is now a glycine, all right? But if this R is a methyl group, now suddenly this amino acid here is an alanine. Now, there are basically 20 types of amino acids out there. And when they form linkages, these linkages is very similar to your amide linkages in production of nylon. But here we call them peptide linkages. Now, why do we call it peptides? Because amino acids, a single amino acid, is also called a peptide. These are the classes of proteins or amino acids. When it's by itself, it's a peptide. When you form short chains of up to 20 peptides, it's called a polypeptide. And when you form like really long chains of peptides, that is what we get finally a protein. A complex three-dimensional structure with really odd shapes. All right? Okay. So, how do peptides form bonds? Well, first of all, you need an enzyme, and then you need to go through what we call a condensation reaction. So a condensation reaction, just like your nylon series, basically says that you're going to have OH lost from one of the amino acids, and it's going to form bonds with the neighboring amino group of another amino acid, and it's going to remove one of the H's. And what you get out of this is an peptide bond. Let me try to draw this, okay? Hold on. H2N CH3, H, C, So this here is your peptide bond, your peptide linkage. Okay, so that here is your peptide linkage. So what happens to these peptides when you form long chains? Well, when you form long chains, now I drew different colored circles or dots to represent different types of amino acids. And these are all randomized because it all really depends on the codon from the RNA, and that's a whole different topic that I'm not going to go down to. So it all depends on what is in our DNA. Our DNA will determine, on top of other many factors, what peptides are being synthesized in our bodies. Okay, it's being made in our bodies. So when you form long chains of peptides, you're going to form what we call a primary structure. It's just a basic, long, simple chain. All right? Now, when these long chains are in our bodies, in our bodies is not a dry environment, it's a wet, complex soup of things. These long chains are going to interact with its environment inside our bodies. And it may form what we call alpha helices, which are things that go round and round and round like that, like a DNA strand. Or it may fold up like a piece of paper in what we call beta sheets. All right? And there's another type of secondary structure that no one really talks about. These are just random structures that just looks complicated. All right? So once you have the secondary structures and you have enough combinations of either 2 or 3 of them, you form what we call tertiary structures, which have a combination of some or all of the secondary structures. All right? And these will form into tertiary structural groups. And when you put all these tertiary structural groups together, you form what we call quaternary groups. And the quaternary group simply is what constitutes the 3-dimensional shape of that protein. And that 3-dimensional shape will help in its function. It is important. You have to imagine that all these proteins are 3-dimensional shape, and that these 3-dimensional shapes determine its function, its usability, how it works in our bodies. Okay? So, just as we eat plant or animal proteins and consume it, our tummies are highly acidified, right? And it's acidified for purely one reason only, which is to break down nutrients. And for proteins, this is gone through a process called hydrolysis, again breaking down this long protein chain to something simpler and smaller so that our bodies can take in those nutrients, right? So in the lab, you can also break down proteins in highly acidified or alkali baths, all right? Now, another way to break down proteins or maybe change its shape is through cooking, and we're familiar with that. When you cook, the food can become tastier or yuckier. That is because you change the shape of the protein. You basically denature the protein, all right? You break down those complex 3-dimensional interactions that is required from one amino acid to interact with the other amino acid in all these complex 3-dimensional shapes. You're basically breaking down its interaction between on its own chain, and now you form something new, a new 3-dimensional shape. And also, you also break down those several these peptide linkages into simpler amino acid chains. And these will add to the character of your food, how it tastes, and its color, its shape, everything that plays an important role. So that's it for proteins. We're going to talk about oils, fats, and soaps.